Thank you, Eddie, for organizing the meeting and for inviting me, and Rafael for collaborating with all that. And I want to thank Monsignor for agreeing to preside over the meeting. Thank you. Initially, I thought that it was a good suggestion that we should have the meeting in Spanish, as you said. But as the meeting progressed, I realized that in spite of the difference, the regional differences that characterizes when we speak Spanish as Peruvians or Argentinians or Mexicans, when we speak English, we all sound the same. And my children, who laugh often at the way I speak English, would recognize us as all coming more or less from the same area. So to talk about the vascular endothelium and in health and disease is actually a very, very wide subject, and it would be very difficult to choose what to talk about. The vascular endothelium, as you know, was a late discovery in terms of the understanding of the vascular wall. It was only discovered in 1845 and described at that time. And a typical endothelial cell is a very flat cell with about 30 micrometers long, about 10 micrometers wide, and very, as I said, thin. And the whole of the endothelium, which is a single layer that covers the whole of the vasculature, doesn't account for even 1% of the total vascular wall components. This shows a typical endothelial cell, and you can see it's characterized by this is polyhedral shape with the cytoskeleton here in green, the mitochondria in this particular slide surrounding the nucleus, and then the nucleus in blue. And the most interesting thing about the vascular endothelium is that until the 1960s, it was considered to be just a covering layer of the inside of the vasculature, that the only role of which was to facilitate the movement of the blood around the circulatory system. And it was considered like a sheet of cellophane. And I became interested in knowing who started to talk about this. And interestingly, the first person who mentioned that was Florey. Florey was one of the co-discoverers of penicillin. As you know, when he reached old age, he developed coronary heart disease and started to become interested in studying the endothelium. And in 1966, he published in the British Medical Journal a very nice study on microscopy of the vascular endothelium. And he wrote there that I have been able to show a little of the current knowledge of the morphology of cells, which 15 years ago were thought to be little more than a sheet of nucleated cellophane. And then he said, I should expect that in the next 10 years will yield a rich harvest of new knowledge about these cells. And he was right. Over the next 50 years, the amount of knowledge that has accumulated in relation to the physiological functions and the potential pathological functions of the vascular endothelium is such that the vascular endothelium is no more considered just a sheet of cellophane covering the inside of the vessel, but a whole organ in its own right, playing a number of roles in relation to the homeostatic regulation of the vascular wall, and also the initiation of some of the early stages in vascular disease. The next slide, just to show you the number of publications that have been generated on the vascular endothelium over the years. This is 1965, 1960, and so on. And you can see in 2010, about 100,000 publications about the endothelium existed. I calculated recently that probably we are reaching the 150,000 publications on the vascular endothelium. And this is just to show some of the key discoveries that played a role in making the endothelium interesting as an area of investigation, and also the relevance in health and disease. The discovery of the mechanism of action, for example, of aspirin-like drugs in 1971. Then in 1973 was the first time that vascular endothelial cells were cultured in the laboratory at Cornell University. Then our discovery of prostacyclin in the middle of the 1970s, the finding of first growth of endothelium-dependent relaxation and endothelium-derived relaxin factor, and then our identification of EDRF as nitric oxide, and the discovery of the metabolic pathway of the biosynthesis for nitric oxide. So there are too many stories to tell about the vascular endothelium and the different roles that we could discussing. But what I would like to do instead is to tell you a story which is a developing story in our laboratory that is, I think, very interesting and starting to elucidate some of the role, for example, of inflammation in the vascular disease. One of the stories that Rafael mentioned yesterday, which is in vogue at these days, derives not only from the discovery of Chance and Boveris many years ago that free radicals can be generated from the mitochondria, but also from our finding in the middle of the 1990s that actually nitric oxide and oxygen interact at the level of the cytochrome C oxidase, and that these two gases interact at physiological concentrations in a way in which is always reversible. And this suggested for the first time that probably nitric oxide was playing a regulatory role in terms of oxygen consumption in the mitochondria, but led to a number of suggestions which are now have generated practically fields of research, because we said at the time that probably if nitric oxide increases in cells, it's possible that there is a decrease in oxygen consumption, a reduction in the oxidative phosphorylation chain, facilitating the release of free radicals and the formation of hydrogen peroxide as a signaling molecule. In the early stages, as a signaling molecule of stress, probably going to become a signaling molecule in terms of hypoxia, but later on, and we demonstrated that there was oxidative stress during this situation, probably reduction in glutathione and the movement from there into a pathological situation in which peroxynitrite was formed and there is true mitochondrial damage. So the idea of mitochondrial damage and the idea of free radical generation through a mechanism related to the presence of nitric oxide has become a very interesting area to follow. And these days it's very popular to think that the mitochondria play a fundamental role in generating free radicals in many different conditions. That's why when I heard about this next story, I got interested in it. And this is a hypothesis which is being is widely accepted these days. It has been discussed many times, and the idea is, and this has been suggested by Brownlee in the United States, that high glucose produces vascular damage, and that increases in glucose in diabetes are the responsible for the vascular damage that initiates the vascular disease in diabetic. And the story more or less goes like this. They say there is increase in glucose. The increase in glucose leads to more glucose entering the mitochondria, increase in the supply of NADPH, leading to some blockade in the mitochondria, probably at the stage of complex 3, and the release of free radicals. Those free radicals then are responsible for the activation of the number of pathways which are known to play a role in the vascular damage not only in diabetes, but in other conditions. Well, from the beginning, I started to think that that's probably not completely true, because first of all, the entrance of glucose into the cells is not completely free. It's regulated by the presence of glucose transporters, and these glucose transporters have high affinity, high affinity but low capacity, and they are operating in physiology at glucose concentrations which are normal at almost full capacity. And secondly, if the glucose was to enter, the glucose doesn't enter directly into the mitochondria. It goes into the Krebs cycle to the formation of pyruvate, which is the one that enters the mitochondria. So there are several regulatory steps that prevent the glucose actually entering, sloshing into the cells and producing vascular damage. And because of that, we decided to investigate whether this was indeed a possibility. There was also already at that stage and exists some controversy in terms of the possibility that glucose produces actually damage in its own right. And secondly, which worried me more than anything else, was the fact that in clinical trials, even a very tight glycemic control does not prevent the development of diabetes-associated vascular disease. So there might be another component, and the idea that we had, and is not a difficult idea to have in terms of metabolic syndrome and diabetes and so on, is that probably inflammation might be part of the whole story, and probably a very significant part of this story. And if this is the case, it would be important to know in which way inflammation plays a role facilitating this process. So we decided to do a number of simple experiments, all in vitro to start with, to try to investigate this. In the first instance, what we did was to isolate vascular endothelial cells and vascular smooth muscle cells, either from surgical specimens or from biopsies, characterizing them and measuring in the first instance as a sign of inflammation the activation of the inducible nitric oxide synthase, which, as you know and you heard yesterday from Rafael, leads to the formation of peroxynitrite, which is a pro-oxidant inflammatory substance. Well, you can put cells in any concentration of glucose that you want for any period that you want, and there is no activation of this system. If you look at the Western blots for iNOS, there is no expression. There is absolutely no expression of iNOS at different concentrations of glucose, and we use 5.5 millimolar as normal, 8, 11, 22 millimolar. And if you measure nitric oxide in the supernatant, there is no increase of the production of nitric oxide. This is completely different if you actually co-incubate the cells with interleukin-1 beta and glucose. And you can see very clearly that interleukin-1 beta produces an increased induction of the nitric oxide synthase. You can see the measurement here in the immunostaining, and the more glucose you put, the more iNOS there is present there. But only if you have interleukin-1 beta. And this is just by immunostaining, showing that 5 and 22 millimolar glucose do not induce the nitric oxide synthase, but in the presence of interleukin-1 beta, it does. The other thing that we measure as an inflammatory pathway was the expression of NF-kappa B. This is we use a reporter luciferase tag peptide, and we measure by luminescence the expression of NF-kappa B. And you can see again that in 5 millimolar glucose, 22 millimolar glucose, there is no NF-kappa B expression, but if you put in the presence of interleukin-1 beta, you do have the expression of NF-kappa B. And this is just the only one slide that I will show you on the vascular endothelium. All the others are on the vascular smooth muscle. This is by immunofluorescence, again, showing that in the vascular endothelium, you require the interleukin beta for the expression of NF-kappa B in the endothelial cells also. Now what happens is that the interleukin beta primes the human vascular cells and the endothelial cells for the effects of glucose. If you pre-incubate with interleukin beta for about 10 hours in the presence of the normal concentration of glucose, and then you wash out, leave the cells for 1 hour, and then put different concentrations of glucose, you can see that the different concentrations of glucose increase the expression of inos. There is a window of opportunity you can see between 3 and 15 hours where this phenomenon happens, and then it tends to disappear. If you put you did the opposite experiment, and you put 22 millimolar glucose for as long as you want, and then you do the washout and put the concentrations of glucose, there is absolutely no priming for this inflammatory response by the glucose to the glucose itself. So what happens is that the interleukin 1 beta, what it does is to augment the expression of the glucose transporters. This is the basal consumption at different concentrations of glucose. This is increasing glucose consumption in the presence of interleukin 1 beta, and this is the expression of glutamate receptors, the increased expression of glutamate receptors when you actually use interleukin 1 beta by immunostaining, and here by measuring of the Western blots. So the interleukin 1, in the first instance, what it does is to increase the transport of glucose. And then the question came, well, if that is the case, is it the entry of glucose the only thing which is necessary to produce damage? If we actually put glucose inside the cell by other means, are we going to have the induction of inflammation? We designed 2 types of experiments well, this is just a summary of what I've been saying. You need interleukin 1 beta to increase the entry of glucose. So we designed 2 types of experiments. First of all, we transfected in normal cells the glucose transporter GLUT1. And the second, and if you do that, you can put the transporter in the membrane. You can demonstrate that the transport of labeled glucose actually takes place. But if you do that, and you put the transporter in the cell, and the cell is not requiring, for any reason, more glucose, there is no increased consumption of glucose. And under those circumstances, the interleukin beta does the effect that I told you, increase in expression of NF-kappa B, but the presence of extra GLUT1 transporters doesn't do anything, as it doesn't do the other way of entering glucose into the cell. We inhibited mitochondria, and if you inhibit mitochondria, there will be translocation of glucose transporters to the membrane. There will be more glucose coming in, and that glucose leads to the activation of glycolysis, but even in that condition, you don't get any inflammatory response. So whether you put the glucose transporters to increase the glucose inside, whether you put the glucose by inhibiting the mitochondrial respiration, you still don't get the effect that you get with interleukin 1 beta. So there must be something else that the interleukin beta is doing. One of the possibilities was that it is changing the metabolic profile of glucose. And we decided to investigate whether there was a change in the glucose metabolism when you give interleukin-1 beta. And we decided to investigate the metabolism of 14C labeled glucose, either labeled in carbon 6, which goes into the glycolysis pathway, or labeled in the glucose 1 that goes into the pentose phosphate pathway, and actually to the carboxylic, but you can, by subtracting, know exactly what you are getting in terms of where the pathway is going. And what we found was that indeed, when you put interleukin-1 beta, there is some increase in glycolysis, but not a very important, although significant increase, suggesting that, again, the idea that mitochondria is the place where the free radicals are generated might not be right. What we found is a very significant increase in the glucose going through the pentose phosphate pathway. And at the same time, we investigated the expression of glucose 6-phosphate dehydrogenase, and there was something very interesting here. Although the interleukin-1 beta was able to activate that enzyme, the presence of glucose seemed to do something very different, qualitatively different, that we still are in the process of trying to understand. So where are we? So what happens is that if you activate the glucose phosphate, glucose phosphate, 6-glucose phosphate dehydrogenase, what you get, obviously, is the formation of metabolites for the synthesis of nucleotides and peptides and proteins and so on. But this pathway is very important for the generation of NADPH. And NADPH, when generated, has two possibilities. Either it activates the glutathione reductase for the regeneration of reduced glutathione to maintain the antioxidant status of the cell, or the possibility to go and activate the NADPH oxidase, which generates free radicals. The Km of the two enzymes are very, very different. Km for NADPH oxidase is much higher than that for the glutathione reductase. But what we found is that when we put the glucose, we lose the activating effect. If we increase the increase in glutathione that we observe when we give interleukin-1 beta, you can see we give interleukin-1 beta, there is increase in glutathione, there is increase in reduced glutathione. And then we increase the glucose, and we lose that effect. And that was the intriguing fact, that we didn't get an increase, dose-dependent increase, but suddenly when you start increasing the glucose, the regeneration of glutathione is not occurring anymore. So the possibility that we are investigating, and is all work in progress, is that once you get to a certain amount of metabolism of glucose through the glucose pentose phosphate, and you reach a certain threshold, you start having NADPH activating the NADPH oxidase and generating free radicals that counteract the antioxidant effect that you get with interleukin-1 beta or with lower doses of glucose. And we found indeed that there is a very, very significant activation of the NADPH oxidase under the conditions of high glucose. So there are a number of experiments to show here in terms of you can block that with an inhibitor of NADPH oxidase, you can block that with an antagonist of interleukin-1 beta, indicating clearly that what is happening at the level of the pentose phosphate pathway. Since there is no more time, let me show you just the summary of what we are thinking about. This is the normal situation. You have low glucose, the glucose entry is controlled, there is the normal activation of the pentose phosphate pathway, maintaining the levels of antioxidant through the regeneration of reduced glutathione. In that circumstance, you increase glucose, and nothing very much happens. Glucose on its own does not produce a vascular damage. We are convinced about that. But if you have inflammation, what you have through the activation of the pathways of inflammation, you have four things that are happening. We are still not very clear exactly in which order and how do they interact, but definitely there is overexpression of glutamate receptors. There is activation of pentose phosphate pathway. There is, of course, in the beginning, activation of the glutathione reductase for the regeneration of antioxidant potential, and of course, the generation of some free radicals through the NADPH oxidase for the activation of inflammatory pathways. If you give much more glucose, then there are changes, substantial changes, increase in PPP activation, increase in the activation of NADPH oxidase, which actually overcomes the protective mechanism that the inflammatory reaction produces. How exactly this plays along and how these things are acting specifically, we don't understand exactly what glucose is doing. Is glucose just synergizing with interleukin-1 beta, or is doing something else which makes a significant difference in all this? The summary of this is there is no inflammation with glucose only. There doesn't seem to be the release of free radicals from mitochondria. Most probably the pentose phosphate is involved, most probably through the series of steps that I have described to you. Thank you very much.